Hi, I'm John Cush. I'm executive editor of Room Now. I'm coming to you from RWCS here in Maui. An interesting week of meetings. I want to tell you about my session with R.D. Kavanaugh called Rheumatoid Arthritis, the Year in Review. We covered a number of interesting topics, and some of the ones that I'll review for you today include the AVERT trial. The AVERT trial was a study that was presented at ACR in 2015, and this is actually a sub-study of the AVERT trial. The AVERT trial was an early RA trial where patients were randomized to receive either methotrexate or methotrexate plus abatacept. The interesting thing in the sub-analysis of the study was what happened to those people who were enrolled with very early disease, meaning a disease duration of less than 3 months. In this trial, patients received one of two therapies, but then at 12 months, all therapy was stopped. The issue being, could you induce disease remission, drug-free remission, how many would stay on? Turns out a high percentage of patients in that trial did ultimately flare and show the need for chronic therapy. But interestingly, in the sub-analysis, it did show that those who had a disease duration of less than 3 months, the response to therapy was faster, and two, for those who stopped their therapy at 12 months, the maintenance of remission or low disease activity was higher in those who had, again, very early disease, such that 6 months later, 33% were still under control compared to only 10% who were on methotrexate alone. An interesting study. Next is two different studies on the same topic, that being polyarticular JIA and what happens when you reduce therapy. One was presented at ACR, another one was actually published in the literature from the UCLA group. The second one, 337 patients with polyarticular JRA who were in remission and then either stopped their TNF inhibitor or stopped their methotrexate. Clearly, and not surprisingly, when you stop the TNF inhibitor, high rate of relapse as high as 80+%. You could, however, stop methotrexate in patients who are well controlled with only a 12% chance of relapse, suggesting that, again, you probably can't go off all therapy. You do need some therapy even when you're well controlled, and it is different as to which therapy you choose to go off of. The SURPRISE study was a study done out of Japan where they looked at patients who were incomplete responders to methotrexate and then either switched them from methotrexate to tocilizumab or to tocilizumab and then maintain the methotrexate. Not surprisingly, in the SURPRISE study, there was a better response in the combination group. So there was a role for methotrexate, even though it was seen that they were not responding to methotrexate alone. These results are similar to the JESMER study reported 2 or 3 years ago, which showed the same thing, but in that study with etanercept. So even low doses of methotrexate should be maintained in patients having an inadequate response to methotrexate, and then when you add on the biologic, not surprisingly, you'll get an additive effect. And lastly was the hot topic maybe at this meeting, because it happened while we were at this meeting, was the FDA hearing on the biosimilar infliximab called CTP13 to be marketed by Celltrion and Pfizer under the name of Inflectra. As you know, it was an all-day hearing reviewing the biosimilarity of the new novel biosimilar to the reference product Remicade, and even the FDA said that this was a highly similar product. After the panel of 24 plus reviewed all the data, they came away with a very positive vote, 23 people in favor of approving that drug as opposed to 3 who were against it. It brings up a lot of issues that still have to be worked out in rheumatology and with rheumatologists, issues regarding how similar is similar. Are you comfortable with just one clinical trial and a host of preclinical trials and validation studies showing the structure, amino acid sequence, binding regions, ADCC experiments, pharmacokinetics, pharmacodynamics? That's the bulk of the work in biosimilar research, and only one clinical trial. Is that good enough for us? And then can you extrapolate that one clinical trial to all the indications for which that drug is approved? The FDA says that once you agree it's biosimilar, then it adopts all the properties of the reference product, the originator drug, meaning it's all of its indications, its dosing, its side effect profile, etc. And lastly, the issue of interchangeability. What happens clinically when you change from the reference product to the new product, or even worse, go back and forth? Is there a downside or an upside to that? That needs to be worked out, and there is no FDA guidance document on interchangeability. And also the issue of interchangeability at the pharmacy level. What will happen when the pharmacist substitutes the old drug for the new drug? We'll see. Watch us for information on Room Now, and come to RWCS, an excellent conference.